Hello. Oh,
Hello, doctor.
good morning. Good morning, guys. I'm gonna start the class, let's say at 8:03, just letting people in. Okay, everyone. Hello. Hope everyone had a great weekend, that everyone's safe as usual. So I'm gonna go and go straight to our lecture. Let me just quickly just share. Okay. I'm gonna turn off my video for the sake of the internet bandwidth. Okay. Okay, can just anyone let me know if you guys can see basically my screen?
Yes, we can see it, doctor.
Awesome. Thank you so much. Okay, everyone. So let's just go back into a small recap of what we were talking about last time. Basically, I think it was on Wednesday. So on Wednesday, we were really talking about how are we able, as let's say, organisms or living beings, to harvest energy. And especially for us, we do that using literally kind of oxidizing. We're literally just oxidizing organic molecules. And we're literally trying to break down an organic molecule into like literally so many different steps. And we're doing it bit by bit. We're breaking it down step by step because we're trying to liberate as much energy as possible. And one thing we're not doing is we're not destroying, let's say, for example, glucose, which is our organic molecule that we use energy from. We're not breaking it down in one step because as we said before, there is too much energy that's released, and some of that energy we can use for work, but a big portion of that is released as heat. So a lot of it is lost. It's entropy. We cannot use that if we burn it in one go. And of course, the entropy, the heat that's released, is so much that it can actually burn us. So we need to be careful with that. So what we do is we do this in sequential steps. And where today we really are going to dive into that. And while we literally are breaking down a big, let's say, glucose molecule into smaller and smaller pieces, we're releasing energy. Every time electron bonds are being broken, and every time electrons are going from one state energy, from a high state energy to a lower state energy is released. So if we go back into, I really like this. For some reason, I cannot go back right now. There you go. This diagram, because electrons from food, every time we pass them from a lower state level to another, the electrons always have to lose energy because they're going from a high state energy to a lower one. So energy is lost, and some energy is lost, as we know, as heat that we cannot use, entropy, and some of it is released that we can actually use for ATP. And every time this happens, we can use this energy to create ATP. And because that's what we use to do energy, to actually, let's say, drive this endergonic reactions that we spoke about in chapter 6. And there's two ways of how we can use this energy. The first one is literally, we call this substrate level phosphorylation. So the name says it. So it's a substrate level, and where we're using the energy from these electrons to phosphorylate. So this really happens, for example, we have a high energy molecule like PEP. PEP is basically something that we've broken down from glucose, which we're going to talk about today. We break the bond so that we can actually use this energy and transfer this phosphate into ADP, adenine diphosphate, to create ATP. So that's one way. And we do this a lot with glycolysis, and where we're breaking down glucose every single time we try to break it down to release energy so that we can use it to actually do substrate level phosphorylation. And we're going to be talking a lot about this today. And during this time, as I said, there's a lot of electrons that are initially being passed around every time. The thing about it is that every step, we need to make sure that we trap the electron because we don't want the electron to be moving around. Because if it moves around randomly and a lot, it actually is extremely dangerous because electrons that are moving around, we call them free radicals. These can cause cancer. These can cause so many mitochondrial diseases. They can cause so many different things. They are actually let's say, center of so many different wide amount of diseases. So we need always to have these electron carriers to accept these electrons to make sure that the electrons are not moving freely so that they're not dangerous, but also make sure that we take the energy and we keep passing it around to release energy to create ATP. And this is called oxidative phosphorylation, in where we're passing electrons from one electron carrier to the other, and we're using all this energy being released because, again, electrons are going from one state to the other, energy is being released, and we're using that to create ATP. Well, this whole thing is called oxidative phosphorylation. I'm going to be talking a lot about this as well today. But just know that for now, during the breaking down of, let's say, cellular respiration, we use substrate level phosphorylation, which we use electrons energy to literally push or kind of phosphorylate ADP to ATP, or we use the passing of electrons to create ATP, and I'll be talking about this a bit more. We'll definitely get into this much more in details. So, and basically most of us, what we use as this, let's say, organic molecule to break down, we use glucose. Glucose is a very great source of energy. And with glucose, what we do is we do both glycolysis, in where we basically are breaking it down to do substrate level phosphorylation, but we're also creating so many electron carriers like NADPH NADH that are literally just carrying electrons, and we can pass these electrons on and on and on to do oxidative phosphorylation. So in a sense, we're literally trying to do both things. This can be literally put into 4 stages. First of all, it's breaking down glucose, hence glycolysis, so glucose. Lysis is the breaking, so breaking glucose. We do this to break down glucose from a 6 carbon into a 3 carbons. Then this gives us pyruvate. So from glycolysis, we get pyruvate, which is a 3 carbon molecule. This then pyruvate is then oxidized further and then enters a citric acid cycle. And where this citric acid cycle, we're just creating so many electron carriers because we're breaking it down more and more and more, releasing electrons. A lot of electron carriers are taking up these electrons, and these electron carriers are then growing and passing these electrons. Hence, you create something called an electron transport chain to create ATP. We're gonna go into details of every single one. Don't worry. So let's start with glycolysis. So this is basically the whole of aerobic respiration. So this is the series where you're breaking down organic molecules, such in this case as glucose, and you're passing the electrons to a final acceptor, which is oxygen, hence aerobic. Remember this, aerobic means that electrons are being passed to a final electron acceptor, which is oxygen. This then creates water. So in glycolysis, so breaking down glucose, as we said, you can burn glucose. You can burn it down to the ground. You release so much energy. A lot of it is released as heat, and a little bit is used for work. We organisms, not just us humans, but animals, insects, plants, you name it. Almost every literally kind of living organism has glycolysis. It's a conserved, basically evolutionary pathway, in where it's a 10-step biochemical pathway, in where we're breaking glucose bit by bit, one after the other. That's what we're trying to do. And this occurs in the cytoplasm. Guys, please, I need you guys to know where these steps are occurring. Glycolysis occurs in the cytoplasm, so inside the cell. And what we're trying to do is we're trying to break down this one glucose, so 6 carbons, into 2 pyruvate molecules. And the pyruvate molecules are 3 carbons. So you're just breaking it down in half, if you want to think about it that way. You do this in a 10 biochemical pathway to create 2 ATP molecules through substrate level phosphorylation. So again, this is basically phosphorylating ADP to create ATP. And you also create 2 NADH from NAD. So because you're breaking down glucose, a lot of electrons are being released, and they're being taken up by NAD, which then is basically, becoming NADH, because NAD is able to take up 2 electrons and a hydrogen. So that's why you get NADH. So glycolysis, as I said, it's a 10-step reaction in where you can actually, let's say, divide this into 3 steps. The first step is called the priming reaction. So this is indicating to your cell, hey, this glucose molecule, I want to break it down. Because of course, you have to always remember our cells don't have a brain. They don't think. You need to really kind of communicate with them. So what we do is we kind of label this glucose molecule with 2 phosphates. This signals to your whole cell, hey, we need to start destroying this. So you start cleaving this into 3 carbon sugars, and then you oxidize it into carbon pyruvate. So we're going to go step by step. So this is what we're going to be talking about. I'm going to break it down into pieces. So before starting, guys, I do not want you to memorize structures. This, I do not need you to know. What I want you to know is the reactions. So that glucose goes to glucose 6 phosphate, and this is basically hexokinase, which gives ADP. This is what I want you to memorize. These over here. Not this, no structures, purely just the reactions. I know it might seem a lot. I remember doing my own bachelor's. I had to memorize even the whole structures. But from us, what you're basically going to be asked in the exam, but also in the MCAT or later on, is these reactions. So I'm going to walk
Doctor,
step. Yes,
uh, I'm sorry for interrupting.
no
Do we
worries.
have do we have to memorize the name of the enzymes?
Yes, you need to memorize the enzyme. So everything literally found here, I need you guys to know it.
Okay. Thank you.
Okay, no worries. Okay, so I'm gonna go step by step, and I've other than this, I have recorded this video before and recording it now, and I've also given literally 2 extra more videos on YouTube, which I've actually used during my bachelor's to work on. So I've given you so much help, and of course, more than happy to explain this further later on if you guys need it. So, as I said, the first reactions over here, the first 3 reactions, what you're trying to do is you're trying to kind of, let's say, kind of signal to the cell like, hey, we need to break this glucose. So what we do is we take the glucose and we add a phosphate. So what happens is that we invest in ATP, we remove a phosphate, so hence ATP becomes ADP, so triphosphate becomes diphosphate, because you've removed this phosphate and you've added the glucose. So this is your glucose, you've added a phosphate now group. You do this with a hexokinase. So the name of the enzyme is really kind of self-explanatory. Hexo means 6, kinase. Just remember, kinase is an enzyme that literally phosphorylates. So every time you hear kinase, it means you're literally phosphorylating. So hexokinase removes a phosphate from ATP to put in glucose to give you glucose 6 phosphate. Great. Glucose 6 phosphate, this is now then turned into fructose 6 phosphate. So what happens is you have an isomerization. So what you're literally just doing now is you're just playing around with its kind of functional groups. You're now creating a glucose 6 phosphate into a fructose 6 phosphate. So you're literally just moving around the whole carbon skeleton. You're creating a different isomer. You're going to see this a lot with these reactions. We do this in order to actually make these kind of, let's say, groups much more accessible and much more easier to break. Because glucose 6 phosphate, it's very hard to break because it's very stable. But fructose 6 phosphate, it's much more easier to break because it is, let's say, the groups are much more together. There's much repulsion. This is what we do. You're going to see this a lot. We're going to do a lot of isomerization. So glucose 6 phosphate is isomerized. So using an isomerase. So you're going to hear this a lot, these kind of enzymes that isomerase. So it's this enzyme is called phosphoglucose isomerase. So it literally isomerizes a phosphoglucose, and you have fructose 6 phosphate. Great. Now we do it very similar to our first reaction. We have a phosphofructokinase. So again, very similar to hexokinase. So it's a kinase. It removes a phosphate from ATP, and it gives you ADP. And it takes this phosphate, and you have a fructose 1,6 biphosphate. meaning that you have a phosphate in carbon number 1 and carbon number 6. So now this is now primed. Your glucose molecule at the beginning now becomes fructose 1,6-biphosphate. It's ready to be literally cut. And it's now cut by an enzyme called aldolase. And aldolase gives you dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So literally these are 3 carbons. So this is this whole. So if you look at basically this whole, let's say glucose molecule or fructose 1,6-biphosphate, it's 6 carbon. You cut it in half to create 3 carbon molecules, a dihydroxyacetone phosphate and a glyceraldehyde 3-phosphate. This is beautiful because this now we can oxidize. This, not really. This is annoying. It's too stable. We don't want this. So what we do is we isomerate it, and we use an isomerase similar to what we did here. We isomerate dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So you have at the end 2 molecules of glyceraldehyde 3-phosphate. This is great. So now we have this 2 molecules of glycerol 3-phosphate. And what you do here right now, now you're entering the phase of oxidizing. You're trying to now break down this even further, breaking down the electrons and taking as much energy as possible. So always remember that when you break down fructose 6-phosphate, you break it, fructose 1,6-biphosphate. When you break it down, you have 2 different molecules, and you're trying to create at the end this 2-glyceraldehyde 3-phosphate. So that's why you need to isomerize this dihydroxyacetone. So now you have the first oxidation. So what happens is literally you have this glyceraldehyde 3-phosphate dehydrogenase. So the name says it all. It dehydrogenases this glycerol 3-phosphate. So you're removing purely. So if you look at here, you're removing hydrogens. So you're breaking down these bonds to release hydrogen. So dehydrogenase. As we know, when you break these bonds, electrons are released. So we always need NAD. So NAD comes up, picks up your hydrogens and your electrons, and you create NADH. So remember, we have 2 molecules. So this is double. So this is done by glyceraldehyde 3-phosphate dehydrogenase. And we also add a phosphate. So here in this reaction, so this enzyme, glyceraldehyde 3-phosphate dehydrogenase, removes hydrogen and incorporates phosphate. So now we have this 1,3-biphosphoryl Now basically we have this 1,3-biphosphoryl. Sorry, we have this G3P, which becomes 1,3-biphosphoglycerate. So it has 2 phosphates. So hence 1,3-biphosphate glycerate. Now what we can do is that this basically biphosphoglycerate has these very rich phosphate groups. So what we can do is we can break it. We break this, and we actually use the breaking of this chemical reaction, which is very exergonic. We can use that energy to phosphorylate ADP to ATP, because now we can transfer this phosphate, and with the help of this energy release from breaking this bond, we release the phosphate, and basically we create ATP. So now we create 2 ATP molecules. And we do this using a phosphoglycerate kinase. So again, kinase is an enzyme that phosphorylates. So we're phosphorylating ATP. So now we have from this 1,3-biphosphoglycerate, we have 3-phosphoglycerate because we've removed the phosphate in carbon number 1. So we have 3 phosphoglycerates. And what we're trying to do again is we're trying to isomerize this again, because now we still have this phosphate group, and this phosphate group can still give us energy, so we can create more ATP from it. Awesome. But as I said, this is too stable. We need to now isomerate it again. We need to play around, change its basically carbons a bit. So what we do is we change basically its carbon, skeleton. So we move up this basically, let's say carbon number 1 up to carbon number 2. So you have 3-phosphoglycerate. This becomes 2-phosphoglycerate because now the phospho is in carbon number 2, not in carbon number 3. So you just moved it around, you assimilated. So you use a phosphoglyceromutase. This is the enzyme to create 2-phosphoglycerate. This 2-phosphoglycerate, we basically remove water from it using enolase to give you PEP. So this is what we talked about before, this phosphoenol pyruvate. Now this is ready. This is super ready to be broken because it's very unstable. So what we do is we use pyruvate kinase. So again, very similar to what we did here with the phosphoglycerate kinase. We break down this phosphate bond, which is very exergonic, and this kinase is transferring this basically phosphate to ADP to create ATP, and you get pyruvate. This is all of glycolysis. It's a 10-step series of biochemical reactions, so one reaction after the other, that this all occurs in the cytoplasm, and you're trying to break down glucose to create pyruvate. So 2 pyruvate at the end, because each pyruvate is 3 carbons. Meanwhile, while you're breaking it, you first invest ATP, so you lose 2 ATP molecules, but you gain 4 ATP and 2 NADHs. So net, so please guys, remember when I ask in the exam, how much are you creating? You say 4 ATPs and 2 NADH. But if I'm asking you, what's the net creation? So what's the total? Well, we lost 2 ATPs, so you're negative 2 ATPs, but you gained 4 ATPs, your net is 2. So please bear that in mind when I ask what's the net. Do not get confused in the exam, okay? So you create 2 NADH, 4 ATP, and net-wise, you create 2 ATPs and 2 NADH from this. And of course, you create also 2 pyruvates. Okay, this is great. You have your pyruvates, but now you have NADHs. The thing about it is that in our system, we have a pool of NAD plus and NADH. Thing about it is that when glycolysis occurs a lot of NAD plus is going to become NADH. So you're going to lose NAD. This is very dangerous because if you deplete all your NAD plus in your body, we cannot accept electrons anymore. And this is extremely dangerous because now electrons are running freely. So we need to recycle NADH back to NAD. We need to remove these electrons and hydrogens to bring back NAD. So this can now still accept more electrons. So we have a big pool of NAD plus. Okay, well, that happens in 2 ways. If oxygen is available, we do aerobic respiration, and where NADH is going to release all its electrons to oxygen. That's where it's going to lose its electrons, the NADH, to become NAD. If we do not have oxygen, this is called fermentation, where we're using an organic molecule as the final electron acceptor. And in our case, we use pyruvate. Because now we've created pyruvate. Well, pyruvate, you're going to become now the final electron acceptor. So so NADH passes its electrons to create now NAD plus, and basically pyruvate is fermented. So the fate of pyruvate, so what happens to pyruvate is very connected to this. If oxygen is basically present, great. We don't need pyruvate to accept electrons. So pyruvate does not need to be reduced because it doesn't need to accept electrons. What we can do with pyruvate is we can oxidize it even further. We can keep taking its electrons more and more and more to get more energy because we don't need it now to accept electrons because we know oxygen will take that. So that's basically aerobic respiration, where we know that our final electron acceptor is oxygen. So now we can actually, keep breaking pyruvate even more. If, as we said, we don't have oxygen, pyruvate now needs to be reduced. Pyruvate now needs to be the one accepting the electrons from NADH so we can create NAD. And this is called fermentation. So this is basically a very complicated, let's say, kind of graph, but it's super easy. So in a sense, now we've basically finished glycolysis. We have pyruvate. Okay? Well, pyruvate, if we have oxygen, which is shown in green, we know that basically, okay, pyruvate, we can keep oxidizing, we can keep breaking it down, and we break it down to acetyl-CoA. So this is literally a 3-carbon. Acetyl-CoA is a 2-carbon. We keep breaking it down. This enters the citrus cycle, which keeps breaking even more and more and more to create more energy. And what we know is that all the NADH that's produced, it's going to go back or recycle back to NAD plus because oxygen is able to accept it. Because at the end of the day, these NADH need to release electrons. They need to be recycled back to NAD because we cannot run out of NAD plus. We cannot. So this has to always go back to NAD plus. So we need a electron acceptor. If it's oxygen, great. We don't need to use pyruvate. We can actually oxidize it and you keep using energy. If we don't have basically no oxygen, shown here in red, pyruvate now becomes your oxygen. So that becomes your electron acceptor. We pass electrons to pyruvate, and this is what we create lactate. So when you basically NADH pushes or gives out its electrons, pyruvate is converted into lactate. I'm quite sure you've heard of lactate, lactic acid. Anyone that does sports will definitely know lactic acid. Lactic acid, this is what basically, when we actually have cramps, muscle cramps, لما انه عضلنا يعضل. Literally, that's what happens when we do a lot of basically, let's say, energy consumption. So we're doing, let's say, we're running, we're going to the gym, and we're basically doing a lot of breaking down of glucose. And a lot of it at the beginning is broken down without oxygen. You produce lactic acid. And lactic acid, it loves to accumulate in your muscle. And this is what gives us cramps. I do a lot of sprinting, and specifically 100-meter sprinting. And 100-meter sprinting, we call this anaerobic, basically, exercise, because you're trying to run in 10 seconds, the 100 meters. That's literally the Olympic standard. In 10 seconds, your body is not able to use oxygen. So what happens, basically, and I feel this a lot, I get so much lactate produced in my body because I'm burning down, basically, glucose into pyruvate. And because there's no oxygen, because it's so little time, I'm creating so much lactic acid. This happens in mammals. This is what we get cramps. In yeast, it's a bit different. Yeast do not get lactate. What happens is that first, pyruvate in yeast, it's removed. CO2 is removed to create acetaldehyde. And now acetaldehyde is the one accepting the electrons. So in mammals, pyruvate directly gets it. In yeast, no. In yeast, you first remove a CO2 to create acetaldehyde, and then it accepts. And you create ethanol. This is how you create alcohol. I love to create, actually, beer at home with my friends. And what we do is we literally try to mimic this whole thing. What we do is we take yeast, we give it sugar, hence glucose. We literally close it in a container that has barely no oxygen. We're trying to starve it from oxygen, so that when it breaks down the sugar, the yeast, it's basically breaking down to pyruvate, which then gives us ethanol at the end of the day. This is literally how you create alcohol, actually, in yeast. So now I'm going to more dive into what happens in basically when you have oxygen, because that's the most, let's say, important parts. So now when we actually get, this case now pyruvate, now when we break down pyruvate, we can break it down, we can oxidize it even further and further and further. Well, that happens in the mitochondria, in eukaryotes. And basically prokaryotes that we know that do not have a mitochondria, this still happens in the plasma membrane. So please remember this. In eukaryotes, when we oxidize pyruvate, this has to happen in the mitochondria. So if we go back here, when we break down glucose, this is all done in your cytoplasm. You break down pyruvate. Pyruvate has to be transported into your mitochondria. Please, please remember this. In prokaryotes, this happens in the plasma membrane. So what we do is we're trying to take pyruvate, we break it down into acetyl-CoA. So we break this down to literally it's this reaction. You remove CO2 to create acetaldehyde, but what you also do is you add a CoA enzyme. A CoA enzyme is literally it's just think about it as a functional group. So now you create acetyl CoA. So you're decarboxylated. You remove a carbon molecule, you create acetyl CoA. By doing this decarboxylation, you're removing a lot of electrons. Well, NAD is going to come up and it's going to take it to create NADH. So in this pyruvate oxidation, you break down pyruvate to a 2-carbon, which is acetyl CoA, because you're literally breaking down pyruvate into acetylcholine. You add basically a CoA enzyme, so that becomes acetyl CoA. And of course, you create NADH. This is literally a series of 3 reactions, or 3 intermediate reactions, where pyruvate is first decarboxylized, you remove a CO2. When you remove a CO2, basically, you're literally kind of oxidizing it. So electrons are being passed into NAD to create NADH. You have basically now this 2-carbon, and you add a CoA enzyme. So it's a 3-step decarboxylation, removing of CO2, and where the electrons, when they're removed, they're being passed to NAD to create NADH. And now you add a CoA enzyme. So this is what you get. This is called pyruvate oxidation. Now, this pyruvate oxidation enters a citric acid cycle. So what you're trying to do is you're trying to break down pyruvate to acetyl-CoA enzyme to further be reduced and reduced more and more. This Krebs cycle is also many different types. It's 9 steps of biochemical pathways divided into 3 segments. It's the creation of citrate, which is 6-carbon. So you take your acetyl CoA, which is 2-carbon molecules, and you combine it with oxaloacetate. And this gives you citrate. And now citrate is oxidized back into, basically, it's literally you're just breaking it down, because you're really trying to take as much energy from as possible. And then the final step is to regenerate oxaloacetate, to recycle this back. Hence, this is why it's called the citric acid cycle, because you start with oxaloacetate and you end up with oxaloacetate. So it's a whole cycle. 9 steps, and this is it. So again, guys, I'm sorry for this. Yes, you need to memorize this, not the structures, but I need you guys to know the reactions. So I need you guys to know, for example, in this citrate synthase, takes acetyl CoA and citrate together, and you basically get isocitrate. So I need you guys to memorize these things, but I do not need you to memorize the structures. That I do not need you to know. So I'm gonna walk this very slowly. So you have your acetyl CoA. This is the product of pyruvate oxidation that we have. So what we're trying to do is we're trying to remove the CoA, so the CoA enzyme kind of group. We're trying to push it out. And we're trying to take this 2-carbon now and add it to oxaloacetate, which is a 4-carbon. And this is all done using citrate synthase. So the name is very self-explanatory, this enzyme. It literally takes acetyl CoA, oxaloacetate, and puts them together to create citrate, hence citrate synthase. Of course, you're breaking down the CoA group. Now you have this citrate. This citrate, it's still, we want to oxidize this completely. But citrate is it's very stable. We don't want that. Now we isomerate this, and you do a 2 steps. Basically, it's 2 steps, which is basically isomerization. You isomerize this, and you do this through acotinase. So now you have citrate. So look at this group. Now you're just changing it. You're pushing it down, and now you have isocitrate. So these are literally basically these are structural isomers. So now this isocitrate is ready to be oxidized. Now we can oxidize it. We can keep breaking it down to release energy. So the first oxidation happens, and this is happening through isocitrate dehydrogenase. So you're removing hydrogens, you're removing stuff from it. So when we remove the hydrogens, what you're doing is that you're passing the electrons, you're cutting the electrons and hydrogens. NAD comes up, picks it up, and it creates NADH. So when you do that, you basically remove the hydrogens and you further decarboxylate it. So you remove carbon. So now this isocitrate, which is 6 carbon, becomes 5 carbons. This is all done through isocitrate dehydrogenase. So you remove CO2 and you also basically bring up NADH. Now you have this alpha ketoglutarate, 5 carbons, and you repeat this again. You decarboxylate it, you remove a CO2. So now this becomes a 4 carbon. You remove hydrogens to create NADH, and you add back up. So this basically CoA enzyme that we lost, we've added back again. So this is called alpha ketoglutarate dehydrogenase. So similar to this dehydrogenase, you're doing the same thing. You're removing CO2. You're now basically removing also hydrogen, so hence NADH has to come up. And you're adding a CoA enzyme. So you have now cysteine CoA, which is a 4 carbon. Because remember, you've now removed basically this carbon. So now what we do here is we try to remove this CoA enzyme again, because this CoA enzyme has a lot of energy. So this breaking this literally reaction is extremely exergonic. So what we can do, we can break this, and what we can try to do is take this energy to do substrate level phosphorylation. So what happens is cysteine CoA synthase comes in, it breaks this, and this energy is now powering GDP. So this is like ATP, but rather than using an adenine, you use a guanine. So now you literally are doing substrate level phosphorylation, and where GDP is being phosphorylated to an organic basically phosphate is going to GDP to create GTP. This GTP is then passing on this literally phosphate to ADP to create ATP. So in this reaction over here, where you have cysteine CoA, you're removing the CoA enzyme to create cysteinate. You're indirectly creating ATP. Not directly, indirectly, because you're now passing, when you're breaking this bond, you're releasing a lot of energy, which you're transferring a phosphate to GDP to create GTP, which then is passing this phosphate to ADP to create ATP. And now you create cysteinate, which is this 4 carbon. Now, what we need to do here is we need to regenerate oxaloacetate again. So what happens is we keep now to do a lot of isomerizations. So here, first of all, we do a dehydrogenase. We do a third oxidation. So we're literally removing a hydrogen, my bad. We do this using cysteine dehydrogenase. The name of the enzyme says it all. So now we're releasing basically electrons. Rather than using NAD+, we use FAD, which is another electron carrier. It's called FAD, and you create FADH2. This is able to accept 2 hydrogens. This is able to accept only 1 hydrogen. This is able to accept 2. So you create an FADH2. So now you have fumarate. Fumarate, now we actually add water using fumarase to get malate. And now malate is remove the hydrogen to create oxaloacetate. So when you basically literally break down, when you remove, again, basically these hydrogens, we pass it on to NAD+ to create NADH. So always remember, every time we have a dehydrogenase reaction, there always has to be an electron acceptor. There always has to be. And finally, you have oxaloacetate. And that's the Krebs cycle. So through this whole reaction, from one literally acetyl CoA molecule, we've created indirectly 1 ATP. We've created 3 NADHs and 1 FADH2. That's our net. So remember, when we have one glucose molecule broken down, we have two acetyl-CoAs. So if I tell you, hey guys, from one glucose molecule, what do you get in the citric acid cycle? Your answer should be 2 ATP, because there's two acetyl-CoAs being broken down from glucose, 6 NADHs, and 2 FADH2. So every acetyl-CoA that enters the citric acid cycle, you get the release of 2 molecules of CO2. So that basically, you reduce 3 NAD plus to 3 NADHs, 1 FAD to FADH2, and you create 1 ATP. And you regenerate oxaloacetate again. This is from each acetyl-CoA. If I tell you from acetyl-CoA, you tell me this. If I tell you from glucose, you double this because you have 2 acetyl-CoAs. Please do not confuse this. This is extremely important. So guys, I'm going to stop here because I know it's a lot of information, but what I've done to make this, trying to help you also from here. So I'm quickly gonna, my computer's a bit slow, sorry about this. Here you go. So if we go here to chapter, this is your Blackboard, chapter 7. I've recorded everyone on its own. So cellular respiration, glycolysis, Krebs cycle. Repeat this as much as you want. But if you guys are sick of hearing my voice, I understand you. I've also added 2 other, basically, let's say, videos that are other people explaining this. But I've also added 2 songs. These are, and I'm sorry for this, these are super cheesy and super annoying songs, but believe it or not, they actually helped me to memorize the glycolysis and Krebs cycle during my bachelor's. So I said, you know what, why not pass this trick on? Of course, these guys are not mandatory. These are for your own help. Use them at your disposal. All right, guys. Is there any questions? Anything you guys want me to go over? We still have 5 more minutes. All right. If there's nothing, guys, please go over it. Do not let this to the final exam. As you can see, it's a lot of things to memorize to these ones. And I'm sorry, it's a lot. I know it's a lot, but use the videos. If you still need someone to understand this, like even despite the videos, feel more than free to let me know. Okay, guys. If not, I will see you on Wednesday. Please stay safe and good luck with everything.
Thank you, doctor.
Thank
Thank you,
you.
doctor.
Thank you, guys.